Thanks. So I'm going to tell you a little bit of work that we've done in my lab. And one of the goals of our lab is really to try to help researchers conduct science in the way that Dr. Sent-Jurgi talks about, basically to look at all the publicly available data and to pull new information out of it and new hypotheses out of it that you might not get through another approach. And because we want to really focus on the new, we're going to try to do this in a way where we're not focusing on prior biology. So we're going to try to have an unsupervised algorithm that can mine these really noisy publicly available data. So if you learned things the same way that I learned things, you would know that this is actually probably an impossible question. Essentially, how do I have an unsupervised algorithm that can pull sort of biological consistency out of data but ignore sort of batch and lab and all these other effects that could otherwise confound an unsupervised analysis? So fortunately, we have students, and our students don't always know that things are impossible. And so this is a student in my lab, Jia Tan, who actually sort of took this project on and ran with it. And so the talk that I'm going to give is basically the story of that work. And so I'm going to tell you how we're taking this sort of machine learning approach and then extending it to try to identify biological networks that are perturbed in an experiment. And this is like, we hope us, basically, giving biologists the sled that they need to go exploring in all their publicly available data. So when Jia was starting in my lab, it was right around the time that some researchers from Google were trying to find what are the recurrent patterns in online videos. And so I assume in this audience, probably everyone has actually, if you haven't read this paper, you've probably at least read a New York Times article about the paper. But does anyone want to hazard a guess at what computers might see if they looked at 10 million images? Cats, yeah. So in other audiences, sometimes no one knows. Yeah, so they see something that looks like this, which, as humans, we can look at this and we can recognize it as a cat. So what are the cats in biology and how do we get at them? So this is the technique that Jia developed. It's heavily based on the denoising autoencoders that are used in other fields, like image processing. And we call this technique here analysis using denoising autoencoders of gene expression, or ADAGE. Basically, what you do is you start with your gene expression data. So in this case, we have samples here. So this is each gene in a sample, and this is red-green coded. Fortunately, it doesn't really matter what it is. But so this would be low, high. And so we have each sample. And then what we do is we add random noise to it just by dropping out the information for randomly selected genes in random samples. And this gives us this corrupted expression vector. And then we train a neural network to recover basically the input data, so the data before we added noise. And what we found is that what we get out of this is a really nice mapping to a lot of biological features in the data that we care about. So for instance, this is microbial data, so we get things like growth rate and strain and oxygen levels in which the microbes were cultured. And so we've been really happy with this type of approach for these data that seems really robust to the types of noise that we see in gene expression data. When I present this in front of computational audiences, I often get asked, so why in the world would you use an unsupervised deep learning style method? Like, why not just go with the supervised classification approach? Because that's essentially what everyone does, and we get amazing classification. And I want to go back to sort of our initial question, which was that we really wanted to extract the biology that we might not know to look for. So we really wanted to only focus on methods that were unsupervised. It was also convenient that while we were trying to get this paper through peer review, LeCun, Bengio, and Hinton, so three pioneers in deep learning, published this paper where they also highlight that they think unsupervised learning might be an interesting application area. It was kind of where the field started, and then the field has moved on to supervised. And so it's nice to hear that they also think that we'll be moving back towards unsupervised approaches. And so I just want to introduce you a little bit to the data that we've been looking at. So this is the publicly available data. I have no idea why random squares are missing up in the corner here. I know why these are missing. So it's noise, yeah. So this is apparently random noise in the PowerPoint presentation about random noise, yes. So each square on here represents 1,000 gene expression measurements that are publicly available, so 1,000 different samples. So right now, when I wrote the abstract, I was hoping there would be 2 million publicly available samples by the time of this conference. It turns out there's not. There's 1.97. So we're quite close to 2 million publicly available gene expression samples. And for this work, we're going to focus on this square here. So this is 1,000 samples. And these aren't just any 1,000 samples, so it's basically the complete collection of gene expression data for Pseudomonas aeruginosa. This is a microbe that's an opportunistic human pathogen. It causes very difficult to clear, or impossible to clear infections in individuals with cystic fibrosis. And just to introduce you a little bit more to this tiny box and tiny window into gene expression, there's actually 100 different experiments here. So if you think of an experiment as something that an investigator did and analyzed probably with a differential expression analysis, published a paper on, and uploaded to Geo or Array Express, that's basically there's about 100 of those here, and they come from many different labs. We tried to quantify the number of labs, but it turns out to be, you can't just use the contact information associated with the sample, because different people upload from the same lab. So we don't have an exact count on this, but at least tens of labs. And so when we applied the ADAGE approach to these data, I'm just going to give you a quick sort of idea of what the result is like. So first, to introduce you to this, when we build these models, what we end up with is for any given neuron, so here this blue circle is a neuron in the neural network, we get a set of high-weight genes. So each of these is a gene measured in one sample, and the high-weight genes are essentially shown here with these thick edges. And it turns out that this tends to follow a normal-ish distribution. It has some slightly different properties, but for all intents and purposes, you can think of it a little bit like a normal distribution. And through a fair amount of work, Gia found that a lot of the genes that are contributing most signal to the nodes are actually these high-weight genes, which is not surprising, right? This is going to be a multiplication operation, so the things that are multiplied by a higher amount are probably also the things that would have the most impact. And if you go back to a set of data sets that are pretty well known, you can actually say, okay, given that I know what I should be changing here, where does the signal come from? And she's settled on a number somewhere around 2 to 3 standard deviations from the mean, basically gives you the genes that kind of characterize a node. It turns out that these high-weight relationships actually capture genes' pathways reasonably well. We're going to use KEGG for this because when we started this work, the gene ontology in Pseudomonas was painfully incomplete. So there were about 152 actual annotations of genes to a function, and that covered 52 genes. And this is an organism that has 5,000 genes in its genome, so it's actually 5,500 genes. So less than 1% of the genes in the genome had any sort of gene ontology annotation at any level of specificity. So what we do here is basically design a quote-unquote protein function prediction method to try to predict genes to pathways, but not with the goal of actually doing a better job of function prediction, but with the goal of asking, does this method extract biological relationships? So this is the simplest function prediction you could possibly imagine with this. So we have a gene here, gene alpha, which ends up connected to node 2 with a high-weight relationship, beta connected to node 1 with a high-weight relationship, and gamma connected to node 3 with a high-weight relationship. And then we have a target gene. So this is a gene that, for the purposes of this analysis, we basically ignored its function. And the target gene is connected to node 1. So we use a similarity, just a correlation of node weights to assign pathways from the closest gene, so it's the single nearest neighbor, to the target. And so when you do this, what you're basically asking is there something encoded in these high-weight relationships that's actually indicative of pathway or function for these genes? And the answer is at least yes, more than random. So if you do this on 1,000 permuted networks, you end up with this distribution, which never gets you more than about 5% accuracy. If you actually look at the accuracy that we observed, which was about 45%, it's well outside that. So whatever this is learning, you could argue it's learning batch effects or lab effects or other signal, but this would argue that at least to some extent it's also learning biological signal because we would expect pathways to be sort of universal across experiments.
This is a straw man, right? Just a correlation based.
If you just do correlation, yeah, so if you just do correlation, it basically just identifies operons. And so operons tend to have the same function. So consequently, if you just do correlation, it gets good performance. If you hold out operons, basically correlation falls apart. And this does not fall apart to the same extent. I mean, it's still, operon signal is definitely there. And so basically, I'm going to sort of do the sort of magic oven approach where we took all the stuff, we put it in the oven, and now we're getting out the complete bit. So I want to talk about some of the ways we can use this for networks. But if you have questions about this, the paper is published. All the source code for this is online. We do go in and show that you can actually identify basically neural network features associated with specific processes. You can then perturb those processes in new experiments where you're specifically trying to change a single node, to change a specific node, and that you actually can change this node. And so we've done quite a bit of experimental characterization to show that signals captured by this are actually biologically relevant and that we can change them in targeted experiments. And so you can read more about that here if you want to. I just want to get into a little bit of the characteristics of the models that we generate. So basically, not surprisingly, if you build a larger model, so you have more neural network nodes in it, you capture more pathways. There's a little bit of a ceiling here. So there are some pathways that we would say probably aren't really capturable either with transcriptional data alone or with the amount of transcriptional data that we have or the diversity of transcriptional data that we have right now. And so in Pseudomonas, these are KEGG pathways. So you can cover somewhere on the order of 80 to 100 KEGG pathways with this type of approach. And there's some interesting kind of performance characteristics. So if we think about capturing pathways, not all pathways are created the same, right? So some pathways are going to be really broad signals that sort of potentially characterize the compendium. So things like oxygen level or other signals. Others are sort of more subtle signals that might be perturbed in a few experiments or might have a more subtle gene expression pattern. And we see this as well. So if you look at something like oxidative phosphorylation, this is essentially how significantly the pathway ends up captured by nodes in an ADAGE model of a given size. And this is increasing the model size. So for oxidative phosphorylation, the smaller networks actually sort of capture it more specifically. And then you can see the signal kind of drops off as you get to larger networks. Conversely, for things like tyrosine metabolism or flagellar assembly, there's basically this pattern where I've added more nodes, I'm capturing more distinct signals, and at some point I get to a number of signals where I can capture these types of pathways. And so it's important to think about, at this point, if you're building an ADAGE model, you really need to think about sort of the granularity of signal that you want to detect. We can also use this to get at networks. So instead of thinking about this as a model where we can identify whether or not a pathway is active in a given sample, we can actually think of it as a way to identify similarity between genes. So thinking analogous to the pathway prediction approach that I mentioned before. What you do is for every gene, you've got these weights, and you can just calculate a correlation between genes for the weight vectors. So basically, how similar are my weights to any other gene? And if you do this, what you get is a network that actually does a really nice job of reflecting function. So in this case, we're looking at similarity in the ADAGE model, and it ends up reflecting functional similarity quite well. So this is a network showing the genes that were most connected in the ADAGE model. We basically took this as a spreadsheet, handed it to our collaborators because the gene ontology annotations were not complete enough, and had our collaborators go in and pick if they had to name one category for what that gene did, what would it be? And so they picked these, I think it's 14 categories along the bottom, as well as the black ones, which are unknown. And so they assigned a function to this sort of spreadsheet of genes. We went back and mapped it onto the force-directed layout, and what we see is that, in general, the genes end up sort of segregating by function. So it looks like, in addition to the kind of analysis we did before for pathway prediction, you also get the same sort of signal here. Going back to the correlation question, if you do this with correlation, because this is a bacterium with genes and operons that are all co-transcribed, you basically just end up with operon balls. And so the largest connected component is essentially the largest operon, because you don't get the sort of cross-operon connectivity. And I think this goes back to something that actually Anastasia found in her talk, which is that if you look at the BioGrid genetic interaction network, the genetic interactions alone give you a certain network, but the correlation over the genetic interactions is actually potentially a more meaningful measure of, at least in their case, function.
I'm having a hard time imagining what exactly is this network capturing that is not, above and beyond gene expression, correlation, and stuff like that. What is it capturing?
So it's capturing gene expression correlation, but it does a really nice job. So if there's sort of really strong correlation, it collapses that pretty efficiently into like a single neural network node. So if you just do a linear correlation, basically that signal ends up swamping all the other signal. In this case, like those really trivial signals just get collapsed. And so it's sort of a sort of dimensionality reduction approach like PCA or ICA, which of course will come up in a few slides.
Another question, if you break down those 1,000 samples into different types of experiments,
yes.
on each one,
yes.
the consistency of your function
So if you break down the 1,000 samples, so I was with you until the function prediction part.
1,000 samples. How
Yeah.
many samples do you need for this?
Okay, I actually took out those slides. Somewhere on the order, it depends how big you want your network to be, but somewhere on the order of about 500 samples, you end up being able to do a pretty good job of capturing things that are like pathway scale resolution, so like the 300 node-ish neural network nodes.
Bootstrap sort of consistency check of these predictions.
Yeah, so we have that. I just took the slides out.
That's good, I'm assuming.
Yeah. So if you want like networks that capture like the 50 largest signals in the data, you need something like 200-ish, 250 samples. If you want to get to like pathways type features, you really need something that's more on the order of 500 samples. So this is thinking. And also going back to mention a little bit more on that too. This is built from a single run of this deep model, and so of this neural network model. And so you can imagine this as like, okay, I've got something hidden underneath. So I've got this stone that's got some carving on it, and it's hidden underneath, and all I've got is the paper on top, and I've basically taken graphite and done like one sweep across that. And so whatever angle I kind of go at, I capture just that type of feature. So we could do this thinking about sort of a stability analysis where instead of just doing one of these, we do a bunch of these. And so we've now done that. So this is the follow-on paper. So my student developed this ensemble approach. So it's an ensemble of these neural networks, which we call ADAGE. Basically, you can train some number of models. We generally training about 100. For each model, you get nodes. You can then identify similarities between nodes within and across models, and then you can collapse those to a single node. And so for the purposes of comparison, we're actually collapsing. We've taken models of a certain size, and then we've collapsed them back down to a number of clusters that matches that. So we can actually do a direct comparison. So you only get, if you get 300 nodes in your individual models, yes, you're going to train 100 of them, so you're going to have 30,000 nodes. But at the end of the day, you have to collapse that back down to 300 nodes for comparison. So it's yeah.
Why ensemble and not just a deeper architecture? That's the whole point here.
Yeah, so we were looking for things that were consistent across different initializations. And so I'm not sure a deeper architecture is necessarily going to give you that. It might even get potentially more sensitive to the initialization.
Well, it's supposed to give you a different level of abstraction over the original assignment.
Yeah, so we wanted the same level of, so we wanted the same type of resolution, but we wanted to focus on the things that were consistent across many runs. So we were really trying to sort of, more like the stability analysis I think Ben talked about yesterday.
When we are at it,
Yeah.
did you vary the number of the size of the hidden layer?
Yeah, so we varied the size of this hidden layer. In the first paper, we used 50 neural network nodes, actually, because our collaborators were going in by hand and annotating because the annotations were so poor. The pathway annotation stuff has let us sort of automate that a little bit. I mean, we know the pathway annotations are not a great way to go because they're just so limited in this system. And so, we use them, but view them kind of skeptically. But yeah, now we're training things on the sort of 300 scale. Yeah.
So if you change the activation function, like what kind of activation function do you use?
Yeah, so this is sigmoid.
So if you change it to like redo, does it have the same kind of, does it pick the same node?
I think it would be a different signal. I think it would capture a different signal. Yeah. Yeah, so we actually like this. So we also like this for a different reason, which is that the sigmoid activation likes to capture features that are sort of generally bimodal or at least multimodal. And so we're interested in potentially one day using these as biomarkers. And so if you have a feature you want to use as a biomarker, the multimodality is really nice because it's easier. You don't have to pick like a cutoff in the middle of a normal distribution for where to draw a line. You usually get pretty clean separation. So yeah, so we've been going with sigmoid, but you can swap it out and definitely capture different things. So basically this ADAGE approach, which if you just use correlation to combine the nodes, it's better than a single model. If you use this ADAGE approach, that, in the interest of time, I will skip the mechanism, the details behind. It's a little bit better. Going back to the initial example of sort of pathways that kind of drop out as you add more nodes, when you do ADAGE, you don't really see this to the same extent. So these are signals that are showing up somewhere across runs, and to the extent that they still end up getting collapsed. We also don't tend to see a performance drop in the things that, that we're getting well captured at the large network size. So we think basically this increases you're basically getting by capturing pathways that are sort of evident at a little bit different levels of sort of model resolution. And the ICA-PCA comparison, since I figured that would come up. So this is, if you just think about sort of how well these things are capturing pathways, again, all the caveats on the challenge of pathways in this sort of not well annotated organism. We see that the ADAGE approach is capturing more of them. We've also gone back and looked, actually in the first paper, we looked at experiments, and we found that if you do the new experiments, you end up with better performance if you're using ADAGE. So that was an ADAGE comparison as opposed to PCA or ICA for reasons I can go into in Q&A if you want more details. But what I really want to mention is that our goal of doing this is not just to catalog the pathways that are there. So there's lots of different ways you can do function prediction and catalog the pathways. So we're really interested in going beyond that and helping biologists basically get at mechanism. So I like this quote from Elizabeth Blackburn. It's basically, I didn't want to sort of build a catalog. I really want to know how things work. And we're really interested in that too. So we've been thinking about how, once you have this model that captures this variability, how do we actually put it to use in the most effective possible way? And one of the things that we've been thinking about is sort of pathway style analysis. So something like Ingenuity pathway analysis, where in our case, we're looking at, so in Ingenuity, you can find like a fold, how much a pathway has changed across an experiment. Here, we're looking at how much a neural network node has changed across an experiment. So you've probably seen volcano plots of fold change. For genes, this is the exact same thing, except instead of genes, these are the neural network nodes. And we use a Pareto selection scheme because we don't like gating on significance. And so we care about things that either change a lot or that are highly significant. Once we do this, if you combine it with the networks, what you get at the end of the day is this sort of pathway style analysis of a new experiment. So this is an RNA-seq experiment where we knocked out the transcription factor ANR and looked at its downstream consequences. It's pretty well known that ANR regulates denitrification, and so basically this green here is essentially the change in denitrification after the knockout. What was kind of cool in this is this quorum sensing group up here. So these are a bunch of genes involved in quorum sensing, and so concurrent actually with doing this analysis, a paper came out talking about showing that ANR actually regulates quorum sensing through these ant genes. So that's good and bad. But it was a kind of nice validation of at least this style of approach. I also want to highlight, just to give you a more concrete idea of the lack of annotations in the system. So what I did in this figure is I actually colored in all of the nodes that had a KEGG annotation in the middle. And so you can, if you look carefully, there's basically these guys up here, which are molybdenum, and then this one NAR gene here. And so there are 5 genes in this that had a KEGG annotation. So if you're using something like GSEA or one of these other methods, your gene set is so limited that you would never pull out either of these signals. And so I think it's really important for these organisms, when we move away from yeast and human and mice, to really focus on how can we develop methods that can work if our annotations aren't there. Because this is a system that biologists actually understand really well. So our collaborator looks at this and right away says, OK, I'm changing denitrification and quorum sensing. So it's not that the knowledge isn't there. It's just that we haven't curated it. And so this is really an area of focus for our group. We also have been looking at sort of where can you do this? So what type of organisms could you build one of these networks for? So if you think about this and looking at, so this is an analysis of Array Express from something like September of last year when I had a grant due. Basically, the number of organisms that have data at that point was around 2,500 different organisms. Now, we can't build an ADAGE model for an organism that has one sample, but if you think about organisms that have 500 assays, which is where we think our method works, at that point, we were just above 150 different organisms. There's no way we're going to get curation communities together for these 150 different organisms to curate to the extent that we've done for yeast and humans. And in addition to methods, we actually build web servers. So we have a web server that we're building for this. Hope to have the Pseudomonas pilot ready by the middle of the summer. And then we're planning to get one together that covers those 150 organisms by early next year. I want to use my last 1 minute to give a shout out to the research Parasite community. So this is a paper from New England Journal earlier this year that branded people who basically do data analysis with data that's not their own as research parasites, which I really hated personally, because this is basically us. Although I would argue maybe we're research mutualists because we actually give biologists the tools to go back and reanalyze their data. But in any case, I thought that it was really important that we, as a community, really maybe own this term. And so I sent an email to the organizers of PSB saying that what we really need is a way to recognize excellent contributions in the secondary analysis, rigorous secondary analysis of data. So I just want to mention we are approved now to have Research Parasite Awards, which I'm calling the Parasites. So if you've seen the Oscars or the Emmys, this is the data science equivalent. There's some selection criteria. The website will be coming up soon. We have a junior parasite, so for a trainee, and sustained parasitism for those who have been around a little bit longer. I just want to finish mentioning that I think the value of research parasites is avoiding the situation that this quote puts us in, which is basically, the things that we really are certain about. This is the chance where we can use data to push back on that, and so we really need to value that. This quote is a perfect example because it turns out it's attributed to Mark Twain, but it's really hard to find any documentation that he actually ever said or wrote it. So it's probably misattributed, which is just beautifully ironic. Okay. So I want to just thank really quickly the people involved in this work. Jia Tan, who's listed up at the top there, is a grad student in the lab who did basically all the ADAGE research. Matt and Dongbo are programmers who are building the ADAGE web server. Collaborators, so the experiments that, the RNA-seq experiment I showed you and all the experiments validating the ADAGE model's biology that I didn't show you that are in the paper were done by Jack Hammond in Deb Hogan's lab. We are shameful users of public data, shameless users, I should say, of public data, so people who follow the rules and put it up there, we couldn't do it without you. Images are Creative Commons other than Calvin and Hobbes, and people who give us money. Thanks.
Ron.
So, first of all, when I just started getting into what is now called bioinformatics, I heard Dan Grauer describing himself as a scavenger, which I think is an even better term.
Yeah, it's a little more positive at least.
Anyway, if you just collapse the operon,
Yeah.
classical tools like clustering, won't you get essentially the same result?
I'm not sure. So you're saying if you basically collapse things that are documented operons, calculate an aggregate summary statistic and then cluster over those?
You're saying the strongest signal is there. Okay, so just take care of it.
Yeah, so I think the analysis across networks of different size suggests that you probably won't get the same answer if you just collapse the operons and then cluster over that. So otherwise, what you would expect would be just, if that were the case, you would expect for all terms like a steady increase in the more nodes you add, the more often you capture that pathway. So I don't think, we did not directly test that, but the evidence we have, I would say, suggests that probably you would not get the same answer with that.
What about comparing to better annotate genome or
Yeah, so actually, the first use of this, so Gia applied it to breast cancer biopsy data back in, actually a couple years ago, and we published it at PSB. I would say, I think basically what we show is, it learns things like stromal infiltration and tumor normal status, and it basically learns all the things you expect it to learn. To me, the value of the unsupervised approach is much higher when you get to these organisms that basically we can't hit with any other method. And so I would say, yeah, we tried it. It was an initial validation, but breast cancer biopsies also had the issue that we couldn't really perturb them well. So then we wanted to go to the use case that we sort of envisioned it being best in. So the thing that's like, an organism that's used to study, in this case, biofilms and other systems, and then sort of really show it like in the situation where we really envision it being used. So I guess I'd say we've done a little bit of it, but then we sort of moved on.
So maybe you said this, but I missed it. Why not do batch correction and then do this if you really are interested in function?
Yeah, so we don't do batch correction because in a lot of cases the batch information is not available. So we're basically just downloading the publicly available data that people have uploaded, and if you take a look at the annotations, so we actually had grad students in Deb's lab go in and try to annotate just some sort of standard stuff across the entire collection of experiments, and it's amazing how sparse that spreadsheet is. So we just do not, I don't think we have the information. I mean, someone somewhere probably has this information in some Excel spreadsheet that's on their computer named like final, something 7. But it's not available to us. Yeah.
So when you talk with your network, when you talk to the human experts, would it be maybe better to also give them a kind of fake network to avoid confirmation bias?
Yeah, so I love the idea of giving them a fake network. So there's a couple ways we could fake the network. So one way to fake the network would be to basically take the genes that were sort of differentially expressed in their study, pull out from the same network a completely different set of genes. So basically randomize the gene expression measurements and do the same thing.
It's like
we
control for that stage.
For the biologist. Yeah, so I would say I've never seen anyone do it. I love the idea. So we're building the web server. So I guess we could do it a couple ways. So one is you put in your results and then you get a network back, and sometimes it's real and sometimes it's random. I'm not sure our users would like that. Yeah.
Real network unless you're the authors of that editorial.
Of that editorial, and then you get it. I mean, I could imagine, so we could put like a checkbox that would basically say, okay, now give me a random network.
So for in the IT,
Yeah.
people have been trying now to add noise data
Yeah. Yeah.
to the validation set. Otherwise, you overfit the validation set too, right?
For IPA.
No, for
Oh,
a lot of the
for a lot of-
tasks, like
IT, yeah.
from Google and things like that. People now actually adding noise to the validation data.
Yeah, so we actually, we do run an experiment in a class that I teach where we give students, I guess this is still on, so all the future students of this class will know what we do if they listen. So we basically give students a data set, and then we give them 2 data sets, and we tell them they need to build a model to predict something. And in one of them, we've randomized the labels just to test over fitting. Yeah, I love the idea of building it into the web server to basically get a sanity check. I think I would do it by randomizing the gene labels. I think we have to give biologists the option to turn it on or not, because otherwise.
Also do different fake ways too. Right?
Yeah.
In general, I think almost 100%, because Keith Baggery from MD Anderson, he does this so-called forensic bioinformatics.
Yeah.
He gave a talk, his talk is on YouTube. He said this, biologists can always find what they want in the data. I mean, it's just, you give them a random set of genes, they're going to say, so he has this example, which I totally believe, said he did some analysis, microanalysis for some lab, and he did it wrong, so the genes are basically random. The first time, he didn't know, so he gave it to the lab. The lab said, oh, this is great, we found a lot of the genes that we know are involved in this cancer, thank you. And then a couple days later, he realized the gene set that he said was completely wrong. He was just really embarrassed. He sent a new set. He said, oh, I'm sorry, the previous set was wrong. This is the new set. No overlap with the previous set. He got an email back saying, this is even better. So I think it's just,
Yeah.
it's so complex now, so that you always can find what you want to see in any set of genes.
Yeah, I mean, so for me, right, this is not like I build a network and then I build a differential expression analysis and that's where it ends. So I would say I think that's where it starts. So then you need to actually, so the ant genes, that wasn't the end, right? That was the starting point for a whole series of experiments testing, if I modify these, do I actually get a downstream change? So, I don't see it as like the end of an experiment. It's kind of the beginning, and that's what we're pitching it for, is more exploratory types of approaches. But yeah, I agree with both of your sentiments. It would be really fun.
Okay, thanks again.